We're going to be talking about the TF binding. So we're continuing on the realm of gene regulation and the DNA regulatory code. And we're also going to be looking at 3D conformational folding. So I'm going to recap briefly the biological foundation from last time. Then I will also introduce three-dimensional chromatin structure. And I'll cover briefly the classical methods for regulatory genomics and motif discovery. And then introduce convolutional neural networks that we heard about for regulatory genomics, and then switch to David Kelley, who's going to be giving us a guest lecture on deep learning and using Bassett, which is one of the earliest tools and one of the most successful tools for deep learning for regulatory genomics. So without further ado, last time we talked about how gene regulation is at the basis of the diversity of cell types in the human body, how this is enabled by compaction of chromatin from the DNA into nucleosomes, chromatin fiber, higher order structure and looping, ultimately to the chromosomes that we know and love, and how this packaging was not just structural to reduce the size of the DNA, but also functional in being able to label the different regions that are packaged up with specific classes of annotations that allow us to now start thinking about the gene regulatory framework that they enable. So we talked about how we can discover these chromatin states based on combinations of histone modification marks. But in many ways, this is the output. This is not the input. The input is the DNA sequence. So a lot of the challenge that we're going to address today is how do we use DNA sequence and how do we use deep learning to predict the different features of the gene regulatory genome? And in particular, how do we distinguish the specific motifs that make up enhancer regions, promoter regions, and also their looping in 3 dimensions. We talked about how motifs are represented by short sequence patterns, and these are going to be at the basis of our convolutional filters. We talked about the hierarchical representation learning that deep learning networks can have in the context of images. And now we're going to be looking at that, in this module, both last Thursday and today. These representations are going to be our motifs, and they're going to be the convolutional filters at the bottom of our deep learning framework. So the lowest level that's closest to the DNA sequence are going to be these motif logos that are going to be learned de novo. And these motif logos are going to be allowing us to now start predicting who are the regulators, how do they combine to predict gene regulatory function. And these logos are basically derived from position-specific weight matrices, PSSMs, or position weight matrices, PWMs, which are these position by position independent models that basically tell you how each position in isolation is dictated by the specificity of binding of each transcription factor. Okay. Everybody with me so far based on the review from last time? So let's see who's with me. Pretty cool. So 653400. So we're going to be learning these parts list, and these are at the basis of much of these, DNA sequence variations underlying human disease. We talked briefly about technologies for profiling those, including chromatin immunoprecipitation, that basically builds antibodies and then pulls down either regulators that are directly bound to the DNA or histone modification signatures that are associated with nucleosomes, and then sequences those to figure out where in the genome every single part came from, mapping back these modifications as to where they're coming from, and then enabling us to now reason about where are these gene regulatory regions in the first place within which we're going to be looking for motifs. So this can basically allow us to now start revealing where are the different transcription factors bound. And we heard many intricacies surrounding that. The layer that I want to add on this is the three-dimensional chromatin structure that allows regions to fold onto each other. Again, just to recap, the cell is enormous, and all of the black in this slide is the cytoplasm. So that's where the proteins get translated, and that's most of the cell. We're now zooming into the nucleus itself, which is the entire blue region, not to be confused by the fact that within the nucleus, there's a nucleolus, which is even smaller. And that's where a lot of compartmentalization happens even within the mammalian nucleus. So if you look at the nucleus, that's where all of the DNA is compacted. And you can see here the different DNA strands that are sometimes touching the periphery. And this periphery is known as the lamina. This is the nuclear lamina. This is basically the boundary of the nucleus prior to getting to the cytoplasm. And a lot of repressed regions are basically stacked up and packed up against the lamina. Then the active regions are pushed away from the lamina and closer to the center of the nucleus, and they lie within chromosomal territories. And this allows spatial organization of the genome that you can actually visualize in the microscope. You can actually see the cytoplasm on the outside, the DNA inside the nucleus, and then this 6 micrometer radius, diameter of the nucleus compared to a 2 meter long DNA. And again, the scale of compaction is quite daunting. It basically spans 9 orders of magnitude between the length and the thickness of the fiber and the diameter within which it's packed. This is like taking a fiber which is between New York and Miami, which is 10 to the 6, which is 1,000 kilometers between New York and Miami, and then fitting it within a 1 meter large ball with a fiber that has a diameter of 1 millimeter. So it's, dramatic compaction. And all of that compaction is happening in stages in various organizational layers. So we understand nucleosomes quite well. We understand how these are made, how DNA folds around these nucleosomes. But then all of these intermediate stages have only recently been elucidated. But we also recognize that there are chromosomal domains where the different chromosomes are actually sitting inside the nucleus. So what we're going to talk about today is a technique for actually inferring what loops to what. How are the chromosomes actually looping on each other? But before I go there, is everybody with me so far as to sort of, the organization here? So let's see who is following me so far. Wonderful. Great. Okay, so we're at 80-25-00. Okay, so now here's a very cool technique for probing chromosomal organization. And that technique is chromosome conformation capture, or 3C. So what does 3C do? It basically says, if I have a soup of spaghetti of noodles that are basically inside my bowl, and I want to figure out where the blue noodle contacts the red noodle inside my bowl of noodles, what I can do is randomly, number one, glue all the noodles together, and then randomly chop up strands at some random length and random fixed intervals. For example, every single time that I see this Hindi3 cut site with AAGCTT, so every time I see this former, I will cut, which basically will cut every 4 to the 4 nucleotides, okay? And that will basically create these random chopped up noodles. I then take a special glue that finds these cuts that are hanging and then ligates them to each other. And that allows me to now create these chimeric molecules that basically contain, part of a blue noodle and part of a red noodle, and then sequence across them to basically see where does the red part bind and map in the genome, and where does the blue part map in the genome. And if the red part maps here and the blue part maps here, it basically tells me that these two were looping onto each other. And if I have a very long chromosomal segment and I see that this part here, is connected to that part there, that basically tells me something about the looping of DNA. Okay, let's see who's with me on how I can use this cutting, chimerization, sequencing through, and then mapping the two fragments to figure out which pairs of fragments, which pairs of regions were in fact proximal to each other in this three-dimensional conformation. It's very cool. So we are at 70%, 20%, 10, 0, and then 4. That's pretty good. Okay, so we can now use this technique to start gaining insight as to how is the three-dimensional genome packaged up. And we can do this at the level of one site versus another site, and that's by using PCR, and that's the original 3C. Or you can use one by all, which is 4C, or you can use many by many, which is called carbon copy chromatin conformation capture, or 5C, or you can use all against all by just randomly cutting and then selecting the fragments that were marked as being recently cut, and then specifically capturing these chimeric regions. How? By adding a biotinylation mark that allows me to, every time a red fragment is cut and a blue fragment is joined to it, every time I have such a junction, I basically have biotinylation, which allows me to now pull down specifically chimeric regions. Because if I don't do that, I'm just going to be sequencing the entire genome. Whereas if I put a little mark here, exactly where they're ligated, I can pull down specifically the regions that were cut. So that allows me to now do this systematically. There are other techniques. For example, ChIA-PET is similar to chromatin immunoprecipitation, but now you're basically doing, again, chromatin immunoprecipitation, but in the end, you do this paired end tagging, whereby after you pull down these regions, relative to a landmark protein, so I want to figure out what pairs of regions is, my favorite transcription factor bound. So what I can do is basically take, I don't know, the CTCF protein, and I can build an antibody against the CTCF protein and pull it down. And then, sort of do the same ligation where I'm cutting up the blue and the red, and then I'm rejoining them. But I can do this ligation through the lens of what is CTCF bound to. And it's one of the factors that's actually involved in chromatin looping. So I can basically use ChIA-PET to pull down a specific regulator that will then allow me to only look at a subset of all of the connections rather than all of the connections. Okay? I've covered an enormous amount of material in a tiny amount of space. So if you're not with me, that's okay. But I'm going to do another poll to see who's with me here. So number one, 3C is basically looking at one region at a time. 4C, slightly more. 5C, slightly more. High C, all of the above. It's basically ligating everything to everything. And then ChIA-PET is only looking through the lens of one regulator at a time. And then there are variations. For example, capture high C allows you to look at only a subset of high C regions, which are associated with things that I care about. For example, promoters. I can pull down all promoters, I can pull down all enhancers of a particular cell type, and then see who they're interacting with. All right, so we are at 25, 55, 10, 10, 0. So yeah, slightly lower, but that's okay. I'm just, introducing a lot of stuff. There's a whole lecture out there that you can watch on the three-dimensional conformation, and we'll post that to you. but this is just to give you a brief sense of those different technologies. All right, so Hi-C is the technology that's the most widely used by adding these biotinylation marks and then being able to pull down these marks and then sequence across these pull down segments to find blue-red pairs that are chimerically ligated. And what this gives you is something like this. What are these pictures showing us? These pictures are basically telling us that this region here interacts with that region here through this 3D conformation. So the genome is basically going down the diagonal, but also down this way and down that way. And every time you see an off diagonal entry, it basically tells you how much is this region of the genome over here interacting with that region of the genome over here. So it basically tells you about loops going this way. Okay, so every one of these pictures you can think of as, you know, looping information between every one of those pairs. Okay, so regardless of what you actually see inside the picture, who understands the coordinate frame of what every single one of these dots represents? So a dot over here basically tells me that this region here is interacting with that region there. So let's see who's with me on this one. Very, very cool. Wow, quite impressive. So 45, 30, 20, 05. Okay, so we can now start asking, well, now that we understand what this picture should be showing us, let's see what it's actually showing us. What it's actually showing us is these, you know, very specific features of long-range interactions. The first thing that we're learning is that very close to the diagonal, you know, regions tend to interact with each other, right? That there's like a thick band rather than just a very thin line. So that's the first feature. There's a lot of, you know, self interactions. The second feature is that there seems to be some kind of boundary here where there's a block where there's much more interaction within the region than across the region. And there's another block here, and then there's another block there. So the second feature is that there are these things called topologically associated domains, or TADs, that basically are much more interacting with each other than with the outside. The third feature is that these TADs appear to be kind of, you know, hierarchical. There are TADs within TADs within TADs. So there seems to be some kind of nested hierarchy. The third feature is that there's this plate pattern. So if I'm this region here, I tend to interact with, and I'm going to call myself, I don't know, you know, kind of like the Jets and the, gosh, I'm embarrassed. I don't remember the rivals of the Jets. But the, you know, there's A's and then there's the B's. Okay. And if I'm an A, I tend to interact more with other A's. And there's another A here and there's another A there. But if I'm a B, I tend to interact much more with other B's. So I'm a B there, terrible choice of colors. So if I'm a B, I tend to interact with other B's. Okay, so there's this checkerboard pattern where the A's tend to interact with other A's and the B's tend to interact with other B's. Who's with me here? So we saw the three features. Number one, the TADs, the groups of regions that are interacting with each other. Number two, the hierarchical nature and how the closer you get to the diagonal, the more interactions you have. And the third one is that there seems to be two types of compartments. There's A compartments and B compartments. Awesome. So 52, 24, 19, 50. All right, so we can now use this to start asking, you know, how is the genome actually organized? We can actually start visualizing some of that compaction. We can actually start asking, what are the features at zoom in and zoom in and zoom in? So basically, across the entire chromosome, you can ask, well, what are these blocks? And then you can zoom into one of these blocks, and then again, you see this hierarchical nature, and you can zoom in further and see this hierarchical nature again, and you can zoom in further and start seeing individual interactions where this region here is clearly interacting with that one very, very strongly. And the rest in the middle is sort of, much more fuzzy, suggesting very specific long-range interactions and suggesting that there's some kind of looping going on between this region of the DNA and that region of the DNA. And you can sort of see this territoriality, these sort of territories, as you zoom in to the genome. You can basically look at all of the different chromosomes, and within each chromosome, you can see these territories, and then you can zoom in further within these territories and find additional territories. Okay? And this is what we are going to call cis interactions within the same chromosome, and these are going to be trans interactions across different chromosomes. So what's really interesting is that many of these trans interactions are now recognized to be reliably seen repeatedly across different experiments. So what you see with this territoriality is that different chromosomes tend to occupy their own space inside the nucleus. It's kind of cool. So basically, this is very similar to the picture that we had seen, very early on, when the different chromosomes were labeled and stained, where you could see that each of these chromosomes was in fact occupying its own territory. And that's what we're starting to see now again, but now at the resolution of base pairs, very, very high resolution. And then the next thing we see is this sort of plate pattern, this A versus B compartment, which basically suggests that if I'm an A, I only interact with other A's, no matter how far they are from me. And what that is suggesting is that there are, in fact, two different parts of the chromosome, of the genome. One part is going to be what we call inactive, and that's actually closer to the periphery, as I mentioned earlier, closer to the lamina. And another part is the active part, which is closer to the center. So there's the A compartment, which is in the center, and the B compartment, which is closer to the periphery. And then I mentioned these topologically associated domains earlier, these groups of regions that are interacting, very strongly within them, but not across them. And you can see those as you start browsing through with this tool, HiGlass, that was developed here at MIT between Leonid Mirny and Nizar, a student that was co-advised by Leonid and myself. And you can sort of see the interactivity here, zooming out, scrolling around, and then looking closer within these regions, and then changing the intensity to see these topologically associated domains as you go through. And then zooming in here, adjusting the intensity again, looking at these regions, and you can see these long-range interactions that are suggesting these looping between distal regions to each other. All right, so let's see who's with me so far. And actually, let's do something following. It's not different. Who feels that they've learned something? Awesome. All right, 27, 55, 14, 0. And then my favorite, nope, I really don't see why the professor is all excited. Okay, so that basically allows us to now start asking, well, what creates these offshoots, these, off-diagonal strong items here? And then what the prevailing model seems to be for these corner peaks, for these loops, is that there's a model whereby different regions would basically come towards each other by a process of loop extrusion. So what people noticed across many different labs is that if you look at the boundaries of these topologically associated domains, you basically would find binding sites for the CTCF regulator, which is sometimes thought of as an insulator protein, but now recognized to be part of that loop extrusion process. So there's a CTCF binding site here, another CTCF binding site here, very often pointing towards each other. And when the two CTCF proteins bind there, they partner up with cohesin, which then pushes a loop of DNA through that loop that they create, and thereby creates these topologically associated domains. Okay, so the loop extrusion model of high C interpretation and chromatin, three-dimensional chromatin folding is that you have a binding site here, a binding site there, that the two regions are brought closer to each other, and then you push the DNA through, sort of effectively growing a loop through there. Okay, so now to recap what we've looked at, we basically saw that there are chromosomal territories where chromosome 6 kind of like to hang out with its buddies, and chromosome 7 with its buddies, and chromosome 8 with its buddies, but much less crosstalk across them. So many cis interactions, but very few trans interactions. And then these compartments of the DNA at 1 to 10 megabase resolution. And then when you zoom in, these topologically associated domains, and then at the bottom, these loops. Okay, so let's see who's with me so far on these 4 different levels of resolution of the chromatin organization. Beautiful. So now let's dive into so 50%, 35, 17, 0, 0. So let's now dive into the computational analysis of these regions. So first of all, how do we understand the language through which DNA is read through all of these different regulators and carries out all of this organization? So the first thing is we're going to be looking for regulatory motifs using traditional approaches. And those traditional approaches before deep learning were about finding patterns that are repeated. So you would basically say, I have a set of genes that are co-expressed, and my hypothesis is that they are co-regulated by a common upstream regulator that binds all of their upstream regions. So I'm going to develop computational methods that look for motifs that are shared between them. And that's what the early methods were doing. They were basically saying, let's define the shared motif using a position weight matrix, which is going to be a latent variable. And I'm going to be iterating between using the matrix to find instances of that motif, and then using those instances to refine the matrix, and using that matrix to refine the instances, and so on and so forth. So basically, I'm alternating between recognizing a common sequence pattern, one of those motifs. In this particular case, it's C, A or T, G, G, C, A or T, C. Okay, so it's a motif, and then I'm discovering the motif at the same time as I'm discovering the instances of that motif. Okay, so who's with me on this key idea of I'm going to be searching for a motif logo, refining the motif logo, using that refined motif logo to find all of the instances, better aligning those instances to each other, and then using the better aligned instances to now define a better motif logo, and back and forth. Awesome. So 50%, 35, 17, 0, 0. That's great. So there are many ways of doing this, but the key idea that I want you guys to embrace, and that's what we're going to be diving in also with David's guest lecture, is that we can use the same deep learning methodology that we've developed so far with this representation learning, with these hierarchical layers, with this de novo discovery of filters. All of that which we initially developed for images, we can now use in the context of regulatory genomics. The key idea is the following. Just like I had my RGB input images for image analysis, I'm now going to have not just 3 layers, or not just 3 channels, but 4 channels. 1 channel for each letter of the DNA. This is what we call one-hot encoding. So the G channel is going to be 1 whenever there's a G and 0 otherwise.
So the G channel only lights up when there's a G. The C channel is 0, 1, 0, 0, 0, 0, 0, 1, 1, 0, 0, 0, 0. And the A channel is going to be 0, 0, 1, 0, 0, 1, and so on and so forth. So I can take my sequence representation, ignore the fact that there are letters, and just simply think of it as an RGB color map where I'm going to have 4 different input channels, one for each letter. Okay? Who's with me here about the bottom layer? So it's a one-hot encoding representation of my DNA sequence that enables me to now think of it as the 4 channels of an RGB image. Awesome. So 74%, 26%, 0, 0, 0. Now, at the next layer, this is where the representation learning becomes super, super cool and useful. The representation learning is going to basically say, I'm going to have a convolutional filter, which I will apply at every position of my entire DNA sequence. Instead of having an edge detector, which I'm going to be scanning through my image to detect edges, I'm going to have a GCATT detector that I'm going to be scanning through my DNA sequence to notice when there's a wheel or when there's a CTCF binding site. The same way that we looked for wheels, for noses, for flashlights, for hats, and so on and so forth. We're now going to be searching for different features of the DNA, and these features are going to be motifs. So the concept here is that we're going to have a deep learning architecture in the same way as before, with layers of representations that are increasingly more abstract. And at the bottom layer, we're going to have the input sequence with one-hot encoding. And at the layer right above it, we're going to have convolutional filters, which are going to be learned de novo. And they're convolutional because I'm applying the same exact operation at every location of my DNA. So who's meeting so far on the convolutional filters? What the convolutional filters are doing are sharing parameters across different regions of the DNA by learning the concept of a wheel or of an edge or of a nose or of a CTCF binding site. And that concept of a CTCF binding site is going to be GATTAC, which I'm going to be scanning through my DNA. Awesome. So we're at 71%, 17%, 13%, 0, 0. That's awesome. So then once I've learned the representation at the bottom layer, and the beauty of all that is that I'm doing this learning, I'm doing this parameter optimization on the fly based on some task. For example, is the transcription factor binding here or not? Based on some task, I'm going to learn representations that will then allow me to carry out the task. I'm going to learn these representations, which are going to be shared parameters across the whole genome. And my deep learning framework is going to realize that the concept of GATTAC is a useful one, and it will learn that motif. And it will learn that the concept of a CTCF binding site is a useful one, and ABF1 binding site is a useful one, and so on and so forth. So it's going to learn all these convolutional filters and then apply these filters throughout the entire genome jointly with a particular prediction task. And once you have that conceptualization, you can then say, okay, great. I can vary the number of convolutional filters. I can vary the length and the size of it. I can vary the number of layers that I want. I can vary the prediction task. I can have all kinds of, input-output relationships. And we're going to see many, many different variations on that theme. Okay? So who's with me here on the overall architecture of a deep learning convolutional neural network applied to genome sequences as a way to extract common properties, common filters, common motifs, and then use those motifs to learn a representation of my sequence, which I will then be able to use for any kind of prediction task. Awesome. So we're at 60%, 40%, 4, 0, 0. That's awesome. All right. So let's do another quick poll. Who feels that they've learned something today? Beautiful. So David, are you there? If so, can you please turn on your camera?
All right, so we're at 60%, 30%, 0, 0, and then 1% always at the bottom. All right. Okay, so David, are you there? Awesome, perfect. So I am delighted to invite David Kelley today. So we are very fortunate to have David. Do you want to share your screen?
Yes.
So David actually is one of the earliest authors of a deep learning framework. And in my view, he has actually developed one of the cleanest such frameworks that truly encompasses very closely this concept of a convolutional neural network and these filters and these representations. So I asked him today to dive right into the foundations of how does deep learning for convolutional neural networks work, from someone who actually invented that and was one of the first 3 papers in the concept. So David, take it away.
Yeah, great. Thanks, Manolis, and happy to be here to walk you guys through this research. And please ask lots of questions. I'm trying to figure out here. It doesn't look like I can see the chat very well.
Don't worry about the chat. We'll be answering the questions, the fearless TAs and myself.
Okay, super. All right, so you guys have had some of the background here on the basics of these models and the goals. So I'll just run through a little bit of intro quickly. So, the overall goal that the research field is really trying to tackle right now is that we know a lot of the principles of how genes are regulated. We know that there's transcription factors binding a promoter that lead to transcription. We know there's distal elements, enhancers that can regulate transcription from thousands, tens of thousands, maybe sometimes hundreds of thousands of nucleotides away, typically by looping. But what we're not so good at is looking at a huge region of DNA sequence, or even a smaller region of DNA sequence, and knowing exactly what it's going to do. We have the principles, but we don't have the precise nature of the language quite down. And then accordingly, when there's changes made to the genome, we have trouble predicting exactly what the effect is going to be on the things we care about. So, this looks like it's a big matrix of nucleotides, or one-hot encoded. We have lots of information labeling these nucleotides. So this is really ripe for machine learning. I mean, me as a human can't make much sense of all these A's, G's, and T's, but I can teach a computer to try to do it. So historically, some of the early successful uses of machine learning on nucleic acid sequences in biology made use of string kernels. So the goal is you have to take this string of nucleotides and you really have to come up with some sort of vector numeric representation of it to be able to work with typical machine learning pipelines. So a string kernel would just say, okay, let me look at all the k-mers in the sequence, thinking that k-mer is an object that is useful. A nucleotide by itself is not very informative, but a k-mer is something that a transcription factor can recognize, that some of the mechanics of the physics of the DNA polymer have relevance to. So by saying, okay, let's count all the formers in the sequence, now I can form a vector, and now I can do machine learning on my vector representation of the sequence, feed it into random forest, support vector machine, anything your heart desires, even neural networks. So the question when I started this was, okay, k-mers have shortcomings here. The simplest one would just be that there's really no easy, straightforward way to take into account spatial positionings of the k-mers. So if looking at a promoter, and it's really only relevant if the k-mer is here, not here or here. That's something that's hard for these kind of bag of k-mers representations to learn. Or if there's any sort of relationships between k-mers. I need this one here and that one there. Well, that's very hard to learn. So, at the time, deep learning methods were really taking off for image analysis. And, it seemed like using deep learning was going to be a potential way to learn better representations of these sequences to make predictions for the things we care about. So the idea here, Manolis sort of covered, but I'll just go quickly through. So we're going to have convolution filters analogous to what people have been doing in image analysis, where you have an image come in and you have these convolution filters that you scan across your image. And the idea here, the inductive bias that you're contributing to your model is that there's translation invariance of the features that you want to extract. That generally, if I see a dog in the upper left corner versus a dog in the bottom right corner of my image, then I want my feature extractors to be able to recognize either of those situations. And so by having these convolution filters and scanning them across your whole image, then you're able to pick up on things like that. So it's the same for us here, except the first layer of the convolution filters are able to basically recognize position weight matrices. That's ultimately what they look like. It's a 4 by the length that you choose object, and we choose it typically to match known transcription factor binding site lengths. And then those are scanning across the sequence. They are creating then a numeric representation that is the number of filters, 3 in this case, so 3 rows by the length of the sequence, which is the sort of element-wise dot product between this position weight matrixy object and the one-hot encoded sequence. Then usually, I'm actually going to plug in a layer that's not on this slide. Usually then we'll do something called a batch normalization. So in terms of how to get these neural networks to really work well in practice, if you want to do multiple layers, there's this operation called batch normalization that is extremely helpful for that. After the batch normalization, you'll apply a nonlinear function. The most common one right now is a ReLU, rectified linear unit, where you basically just set all the negative values to 0. So we just want positive recognition of our position weight matrices. We don't care like how much a certain string doesn't look like our filter. We just want the occasions where it does look like the filter. And then ultimately, if you want to be applying this multiple times, then you typically want an operation that starts to summarize and aggregate information. So here, the final step, this max pooling operation says, let me look at adjacent positions, and then for each filter channel, I'm going to take a maximum operation. So I'm just going to send through the maximum value. And so that starts to code things like, well, if I recognize a transcription factor binding site, I don't so much care if it's at position 12 versus position 13. That doesn't make a huge difference. I might care about position 12 versus position 800, and that's fine. That's why the max pooling only covers like a little small window. Okay. So stitching that together, we come in with our one-hot encoded sequence. We apply this convolutional layer followed by a max pooling. Then we can start to repeat that operation. So if we just did one layer of the convolution, then it starts to look like, okay, maybe I was just using position weight matrices and just looking at their output. And that's not very in tune with the philosophy of deep learning. So instead, we continue applying convolutions. In the model that I'm presented here in the original paper, I did this 3 times where I did a pooling operation, and then you continue on. So the second and third layers no longer have this sort of easy interpretation because the convolution filters don't align with transcription factor binding sites like the first layer. Instead, what they're able to learn is, okay, they're seeing as input a vector of all of the first layer's filters and what they found. So the second layer can learn things like, oh, well, this filter and that filter activated, so now I'm going to activate. So if there's any sort of relationships between the transcription factors, or if there's a relationship like I need to see a transcription factor binding site, and then I need to see some GC content richness. then the second and third layers can start to learn those sorts of relationships. Finally, you want to come down and make a prediction. So after they've done the convolutions a few times, you still have this object that is the depth of your filter channels by the length of your sequence. And in this case, I just wanted to make one prediction for the sequence. So I need to collapse across the length axis. So the way I do that is just flatten. So now you sort of just kind of thrown away the positional information, and you run a now fully connected layer, like a multi-layer perceptron, and you make your final prediction. So in this case, I'm going to bounce ahead and show you the data and then hop back. In this case, the data that I wanted to predict was DNase hypersensitivity, which measures the accessibility of a region of DNA on the order of 100 nucleotides, 100, 200 nucleotides. And what we had access to, thanks to incredible efforts by consortiums, to which Manolis is a major contributor, is tons of data like this across many different cell types. So what I focused on was a curated set of 164 different cell types. That's the rows in this matrix, and binary calls for whether there's a significant signal from this DNase hypersensitivity assay measuring the open chromatin. So to say just a little more about the DNase, the simplest way to think of it is combining all of the binding of different transcription factors. So, there's going to be lots of different motifs that are determinant of whether there's a DNase hypersensitivity peak. And you can see from this matrix that although there are a bunch of sites that are pretty consistently accessible across cell types, there's also lots of cell type specific peaks. And we want to be able to learn those. I mean, those are potentially more interesting to some biologists. Some of the consistent ones might be promoters that we know would be accessible, but the cell type specific stuff might be more interesting. So there's about 2 million sites. We're going to break them up into a training, validation, and test set. We're going to train on a bunch of them. We're going to test on the others. Okay, just to hop back now. So we have these 164 different cell types. So the final layer here where we make a prediction, we're going to output 164 different predictions. Now, it doesn't have to be that way. You could just take each of these experiments one by one, and you could train an entire model to predict just that one experiment. But it turns out that even though there's a lot of cell type specific stuff, there's enough structure and there's enough sharing of motifs and there's enough transcription factors that are consistent across all the cell types that you get better results if you do it in a multitask learning setting. So multitask learning is, okay, I have lots of different prediction problems and I'm going to combine them into one model. And then you, the practitioner, have choices to make about how much to share between each task. But here we're going to do a pretty strict sharing. So all the convolutions and the fully connected layer are shared between all the tasks. The only parameters that each task has that is specific to that task is just this final linear transformation. So
can I jump in to tie it a little bit with the lectures that we've had? We talked a lot in lecture 6 about representation learning and how you can actually trick the deep learning framework to learn representations by effectively giving it dummy tasks. In this particular case, these are not actually dummy tasks. These are a lot of very useful tasks. But what you could have done is trained one deep layer, one deep convolutional neural network for one task and another one for another task and another one for another task and another one for another task. The downside of that would have been that your internal representations might actually have been slightly different. That basically maybe this task is great at recognizing 4 transcription factors, and that other task is great at recognizing a different 6 transcription factors. And a third task has 2 of the first, 1 of the third, and another one that hasn't been seen before. And suddenly you'd end up learning a lot of different variations of these underlying regulatory motifs, as opposed to forcing it to have one task all of the tasks jointly, and therefore one representation that's joined for all of these tasks. Again, the trick is to get the deep learning model to learn representations of the underlying meaningful features of our data set, which in this particular case are the motifs, and those motifs are very often shared and reused across many of these tasks. David, what are the tasks that you're doing here? Is it just different cell types? Is it different TF chip experiments, or any of the above?
In this case, in the original paper, it was just different cell types with the DNA's hypersensitivity assay. But in follow-up work, I've included any data from the functional genomics world that generates peaks that can be represented in this format. So in that case, it can be transcription factor ChIP-seq, histone modification ChIP-seq, ATAC-seq, trivially is quite close to DNAs. Anything that you could represent as like a punctate binary peak.
Great. I'm going to do a quick poll to see how well folks are following. So let's see. Can everybody sort of understand this multi-layer architecture of Bassett and how it maps to everything we've learned before? Great. So 41, 41, 18, 00. Awesome. Take it away, David.
Great. I see a question here in the chat that a question about the x-axis. I'm going to guess the question was from this slide. Here, the x-axis is position. You know, it's position across the sequence. Okay, so you know, at the time, I showed you some of these Kamer SVMs. At the time, the Kamer SVMs were the most competitive models for these sorts of tasks. In fact, there's a model called GKM SVM that's using gapped Kamers. It's really an excellent model and still widely used today. But in a task like this, you're able to get better performance on the test set by using the convolutional neural network, which is it's just a more flexible model. It can represent more things than the Kamer SVMs can represent. Okay, so wanted to get in next to try to understand the model, figure out exactly what is it doing, and you know, this is useful as part of the exercise as a practitioner because typically you can identify something once you understand the model better and go back and iterate and maybe change the architecture and change different parameters of it. You have a lot of choices when you do these sorts of convolutional neural networks. So the better you understand your data and your model, the better you can set those parameters. So trivially, the convolution filters, you know, we pitched the idea that they would be representing transcription factor binding sites. So first we wanted to check if that's true. So the filter objects themselves are just continuous real values between, you know, typically end up between like -5 to 10 or something. So to figure out what they're recognizing, I would take the filters, scan them across a whole bunch of test sequences, figure out which places in those sequences the filter activates, and take the highest activations and build a position weight matrix out of it. And then I can compare those position weight matrices to the databases we have of transcription factor binding sites. And the one I'm using here is called CISBP. And I can see that indeed, this filter from the Bassett model recognizes a sequence that's extremely similar to the database motif. So that's really promising. And if we look across lots of different convolution filters, that's largely true. I should say that this is a little finicky. It doesn't have to be this way. And there are certain settings of the architecture that make it more or less likely that you'll learn these very position weight matrix filters. And, you know, so that's just something to watch out for. Another piece of sort of complexity to it is that for position weight matrices, for transcription factors that are really useful to the model, it's not just going to use one filter that would match the database. So CTCF is a really important factor for predicting accessibility. And there's many different filters throughout the Bassett model that recognize something that looks like CTCF, and I kind of line them up here. So that's interesting because it indicates that.
Can I ask you a question? So
yeah,
if you had additional layers, basically what Anshul talked about on Thursday is that with additional layers, higher order layers, he would end up sort of combining two sub-motifs in a larger motif. My question is, did you look into sort of potentially, I mean, because CTCF is an unusually long motif, and maybe the individual components of it, I mean, it has multiple fingers that are sort of touching the DNA. My question is whether having multiple layers would allow you to perhaps combine more building block representations of it.
Yeah, definitely. It's in the first paper, I was lacking in the creativity to sort of quantitatively establish what the deeper filters are doing. Yeah. After seeing some of the work Anshul's done, kind of gone back and said, okay, well, even if it's a deeper layer of the network, you can look at what's activating it. Go back to the sequence and see what's active.
Perfect. Great.
So you can definitely do this sort of same exercise for deeper layers and see, oh, it's recognizing a fuller version of the motif.
Thank you.
Sometimes it's tricky because it'll combine the forward version with the reverse version, and now the sequences are sort of bimodal. There's like two different types of.
So how do you avoid that?
So you can imagine, like if the orientation of the motif doesn't matter for making predictions, then at some point in the model, it's going to combine recognition of the forward and the reverse. And
another way might be for the pooling layer to basically search for motifs in both directions.
Yeah. And there's ways to take advantage of this in a more clever way, and Anshul's written some papers about that. And it seems to help when you have small data because you're imposing a stronger inductive bias.
Exactly.
But when you have a huge data set, it seems to not matter too much. You just have enough information to learn it all, and you get by.
Great, thank you.
Yeah. So the other thing to note here is that this is good. There's lots of filters that recognize it. If there's variant versions of the motif, if the position weight matrix is insufficient to represent this transcription factor, then maybe having a bunch helps us make better predictions. But it makes it a little hard to say exactly, where is CTCF binding and how is CTCF contributing to the predictions, because now the CTCF information is now spread across many different filters, and I sort of would have to keep track of that. So I tried to do an analysis that sort of bears out this challenge. I took every filter, and so in the scatter plot here, these are all filters. You can sort of see some of them colored. And on the x-axis, it's the information content of the filter. So CTCF has very high information content, so it would be over here. But some of these have very low information content. This one just recognizes a CG, really, so that's low. On the y-axis, I'm looking at the influence of the filter as quantified by getting inside the model when it's trying to make predictions and setting the filter output to its mean. So now there's no useful information coming through that filter. So if it's really important, then the model will be handicapped and it won't make good predictions. And that's what this influence measures. And I made this plot and thought, oh, CTCF is going to be highly influential. And the problem is it's not, because there's no individual filter, because you could knock out filter 200 or filter 106, but there's still 7 more that can be used to make those CTCF predictions. So it's very robust to the loss of one filter. And the filters that end up being the most important are these sort of picking up on nucleotide composition information, GC richness. So that sort of gives you a taste of some of the challenges we get into with interpreting these models. Okay, I'm gonna skip through that bottom part. if you do have transcription factor motifs that you know are really interesting to you, there's one more analysis I did that I think did work pretty well. So once you have the trained model, you can take any sequence you want and give it to the model, and you can take modifications of that sequence and make predictions. So you can take this known motif you care about and just start injecting it into other interesting sequences. So here I just took 100 random sequences from the genome and put in this transcription factor motif for the AP1 complex. And I can see that in different cell types, there's a different reaction to that motif, indicating that there's some cell type specificity. You know, that this brain region here didn't seem to care too much about it. The stem cells care a little, and the HMFs over here care a lot. Most of the sequences go to very high accessibility predictions. So that's one way you can start to study transcription factor binding sites. Another way, which it sounds like maybe you got a taste of from Anshul, is to come into a sequence of interest and mutate every single nucleotide across the sequence to its alternatives in order to see which mutations to the sequence induce a change in the prediction I want to make. And you know, there's alternatives to this that Anshul has pushed forward, where you're making use of the gradients, and you know, there's pros and cons computationally to either of the approaches, but they're trying to capture the same thing. What is the influence of this nucleotide on the prediction? And when you do that, you see these motifs start to emerge out of the sequence. So over here, you have this AP1 motif where mutating any of these nucleotides is going to significantly decrease the prediction. And in the paper here, we verified that, you know, there's ChIP-seq for this transcription factor at that site, and there's some reasonable conservation statistics on the motif, sort of helping to validate that that this is a pretty good labeling of those nucleotides. Okay, so you know, at this point, I thought that we had a pretty reasonable model for looking at a region of DNA. The peaks, like I said, are about 100, 200 nucleotides. I give the model something like 600 or 1,000 nucleotides, so it has a little more context. But like Manolis presented, you know, there's really long-range correlations in the genome, and we'd really like to be able to take better account of those. And more specifically, you know, we want to be able to predict transcription and gene expression. You know, those are ultimately, all these transcription factor binding sites and chromatin data is sort of on the path to the expression of genes. And if you have a mutation and it just modifies a transcription factor binding site that affects no gene expression, then you probably don't care about it. You want to zero in on the ones that actually change RNAs and ultimately change proteins. Okay, so we know a lot about the principles of how this works. Like I said before, we know there's promoters, there's enhancers, they have their transcription factor binding site motifs in there. We know something about the 3D structure, and there's these insulator elements with C2CF. So can we look at this broad region and use these techniques to compute a function of all these elements to predict the expression of this gene? That's the problem that I took on next. So I'm going to work with the same sort of bones here of the models. We're going to use the same convolution structures as before. The differences are, we're going to bring in a much larger region of sequence. So in this next work, I'm bringing in about 100,000 nucleotides into the model rather than 600. So much, much more. And then after we do our convolutions, we're going to do the max pooling. We're going to get the sequence to about 128 base pair resolution. So then across this long region of sequence, we have vector representations. And after the convolutions, it's going to roughly know, like, okay, this looks like a promoter, this looks like an enhancer, this looks like a C2CF site. So the challenge we face next is, how do we share all this information across the genome? How do we let this promoter know that there's an enhancer over here and another over there, and maybe it's blocked by this insulator over there? We need all these elements to know about each other. So there are several different modules you could plug in here now. When I first started the project, I hypothesized that recurrent neural networks were going to be the best tool for the job. So I was going to have a recurrent neural network that would step across the sequence. It would say, aha, an enhancer, aha, a promoter, let me update because I saw an enhancer, and then keep going across the sequence. The problem I ran into was that the RNN implementations a few years ago, this might be better now, were just too slow on these really long sequences that I wanted to bring in. I was just having to wait forever to train these jobs, and it just wasn't practical. So instead, I turned to another tool that had been demonstrated in machine learning papers to be useful for this task, which is called a dilated convolution. So I'm going to hop ahead and then I'll hop back again. The dilated convolution idea is, let's stick with our convolutions, scanning across the image for the convolution, but let's dilate it in that we're going to expand it and we're going to insert gaps into the convolution. So on the left here is just a normal convolution, and we'll call that dilation rate 0. Over in the center, we'll call this dilation rate 1. So now between every 2 positions we actually look at, there's a gap. And in 2 dimensions, it kind of expands the filter in this way. And then the next one is dilation rate, maybe you'd call this one 2 and that one 4 or something. At any rate, you're kind of continuing to expand the size of gaps between each position. Okay. And you can do this by stacking these and growing the receptive field, the number of gap positions between each position exponentially. You can cover the image very quickly, very parameter efficient. And you can maybe imagine how this would be very useful for tasks like image segmentation, where you have your convolution captured representation of each region of the image. But to really segment it well, they all need to know about each other. So you got to pass all the information around before you segment. So stepping back here, the dilated convolutions, we're going to start small. We're going to quickly expand. And then if you look at this bottom one here, you see that, okay, now we're doing a computation with this convolution that looks at the center position and then looks at positions that are quite far away. So even though this one is hitting here, which is kind of a boring position, really we wanted to look at this enhancer position. The key is that this enhancer, this boring position here has had opportunities to communicate with this enhancer here. So the information is kind of slowly getting spread around. And even though I'm hitting this position rather than this one, there's still an opportunity for the information to pass through so that the promoter can be notified that there's some interesting regulatory elements around. And then ultimately, similar to before, I'm going to make predictions in a multitask framework. The only difference here is I'm going to maintain my length of the sequence, maintain the length dimension, and I'm going to make predictions in bins across the sequence here. So every 128 base pair position, we're making a prediction. And for the DNA hypersensitivity, they're very local. You got your ChIP-seqs, and then CAGE is a way to measure gene expression or RNA abundance that is centered right at the promoter. So this is a nice assay for convolutional neural net learning because all of the information about the gene's expression is right in the promoter, where the convolutional neural net is best able to find the sequence information that determines that gene expression. So we're going to predict all these chromatin data as well as the gene expression. Okay, one last note. So you may have heard, if you've been following the deep learning, that it was realized a few years ago that residual connections or skip connections in the neural network can be really helpful for training deep nets. So once you want to go to like dozens or even hundreds of layers, you really need these residual connections. So what is that exactly? Well, it means that for every layer, the input matrix is passed directly on to the output. And after you do your convolution, batch norm, ReLU, whatever you're going to do, you take the output of that and you directly add it to the input. So now the difference between what we were doing before each layer needs to sort of relearn all of the information and pass it all on. With a residual network, each layer just has to learn new information because everything that's already been learned is going to be directly passed on. We don't have to sort of reinvent the wheel. And that's nice for these dilated convolutions because each one is looking at something different and further away. So we're saying each time we look further away, let's just learn something new and let's just directly pass on what's already been learned with the residual connection. So that's ultimately how, in the first paper, if you go back and follow this reference, I'm not doing that, but I recommend doing that. I realized after that it's a better way to set things up.
David, can I jump in just a little bit here? So
yeah,
the residual network connections are usually done in the field across different stacks. But what you're doing here is across different genomic positions, right? So it's more residual information and residual networks on the RNN dimension rather than the CNN dimension, right?
Yeah. And it's analogous in the image world in 2Ds. Each 2D position would have its vector, and the residual connections would happen there. At each 2D position, you'd learn something new and add it into the vector. Here, it's analogous. It's just that we have a 1D rather than a 2D.
And the cool thing here is that because you're adding it up and because you have these sort of dilated networks, you're effectively bringing in the raw information and the parsed, quote unquote, information from both near and far, right?
Yeah.
Beautiful.
Yeah, that's the idea.
Awesome. Let me do a quick poll here to see who's with us. So can everybody see these sort of dilated networks and how you're dilating across genomic positions to bring information closer at various levels of expansion, and then combining both the raw sequence and the sort of higher order information using this residual information, which I think will be a good thing to cover in recitation as well. Awesome. So we're at 37, 32, 26, 50. So yeah, definitely worth covering. All right, David, continue.
Great. I see a question there asking if the raw input is concatenated to each filter. And so a couple things. So there's the raw input in that there's the nucleotides at the top layer. Those are gone once we do. Sorry, step in animation here. Those are gone once we start doing convolutions. So for the residual layers, it's the input to that layer that gets passed on, not the input from the beginning or the raw input of the nucleotides.
Perfect.
And then to clarify, because you asked, is it concatenated? No, it's not concatenated. Well, I probably confused you because of this setup here. But in this case, no, it's not concatenated. It's an add operation. So, when you're doing your PyTorch or your TensorFlow program, you have your input matrix, you do your convolutions, and then you add it in to the input to that layer.
And is that adding problematic because you don't have either of the two, you only have their sum?
No. I mean, I think, the idea is that the input, like I said before, the input has learned all this information. And so by just adding it on, you're kind of updating each position's vector by taking into account this new information. And then after you do the add, then people often do another normalization, maybe throw a convolution on top of that, so you can start to really get things fixed. Another question, how many residual connections are there? So that kind of depends on the length of the sequence you want to work with, because the most important thing is we want to allow those dilated convolutions to be able to look far enough, but not too far, because at some point, if I kept going here, it would be the case that I'd start to hit outside the bounds of the sequence a lot. And at that point, you're not really going to learn anything. It doesn't hurt too much, but you're not going to learn anything. So usually I'm doing something like 5 to 10 dilated convolution layers for an input sequence of 100,000 nucleotides. But that would change as you scale the sequence, or if you change the bin size. You just have to sort of work out the math and figure out what you want. Okay, so the input in this case is the continuous signal from all of these different data sets. And the reason I had to switch to that is that the cage, the gene expression, can't easily be binarized. You could, but you'd be throwing away a lot of information. And it turns out a lot of the assays sort of have that property. They can be binarized and it's fine, but there's a little more information in the continuous signal. So we're going to predict the continuous signal here. We're going to use you can use a mean squared error loss function, that's fine. But all the data is positive and it's derived from counts. So I'm going to use a Poisson loss function, actually. That's what I found to work the best. And you feed it this region of DNA. Here's the experiments. This is a held out test sequence, and we're able to recapitulate the patterns for most of these assays. So that's good. There's lots of different assays in there. There's sort of different accuracies, our ability to predict them. This is a little hard to interpret. The way I set it up here is the x-axis of these plots is the correlations between replicates. So some of the assays are just noisier than others, and that determines the quality of the model that you can get back. On the bottom right here, for example, the H3K9 methyl, some of these have low correlations between replicates. It's a tough ChIP-seq to execute, and so the quality of the models is a little less. But when you have really high replicate correlations, like the H3K4ME3, this upper end, you can get very high quality models. But I think this is an important principle to leave with, that for all the choices you make in your deep learning model, ultimately one of the most important factors to the quality is the input data coming in. And the field is continually improving these assays and moving to finer resolution things, and that's going to be great for these sorts of models. Okay, so Manolis, we just have 6 minutes left, right?
Well, yes, technically.
Okay. All right. Let me just quickly think about what we're going to cover. So maybe I can stick with finishing the stuff here and maybe skip the high C. Do you think that's right, or should I jump ahead and cover the high C real quick?
I would say tag the high C in the context of this one. I would, yeah, I think it's helpful to mention it briefly for a few minutes.
Okay, great. All right, I'll just briefly mention that again, like I did some analysis with this model. In this case, making use of the gradients. You can look at the gradient of the prediction with respect to the nucleotides. We talked about that, or you can take it with respect to any of the intermediate layers, and that was useful for sort of labeling regions of the sequence that the model was making use of. I'll just leave it at that. And then, ultimately, we love to be able to make predictions for mutations. So there's a lot that you can do there. For example, this is a disease-associated SNP, and you can dive in and see that the nucleotide is part of the transcription factor motif, and it affects predictions and cell types that are potentially relevant to the disease. And this is a major driver of the reason we do these analyses. And now that I work at a company, it pays the bills to be able to connect this sort of basic computational biology research to disease associations. Okay, so Manolis covered the principles of 3D regulation in the nucleus, that the chromosomes are folded up and very compact. And this folding is quite important for gene regulation because it determines which sequences, even if they're far in linear space, are going to be near each other in three-dimensional space, and are better able to sort of share transcription factors and share regulation. So the question I had with collaborators who've been really working intensely with high-C data and these sorts of models was, can we predict the 3D contacts derived from the high-C experiment from the underlying DNA sequence? So the way we're going to go about this is we're going to take the high-C data. So starting on the upper right here, this is the object that we want to predict. And it's two-dimensional. So you have nucleotides across the x-axis here and nucleotides across the y-axis of the matrix. And each position is the contact frequency between that bin in the genome and another bin in the genome. And there's a really strong distance decay in this data. So we're going to do a transformation where we normalize out that distance decay. And what we're actually looking at is a kind of a log observed counts over expected counts given the distance decay. And then I'm going to use a mean squared error. So I want to predict these objects here. These.
And what are the multiple layers of these objects that you're showing?
The multiple layers is multiple cell types. So again, we're going to do multitask learning. There's a bunch of high-C experiments that are high quality enough to predict. So each of these is a different cell type high-C assay. Okay, so the model we're going to use has to get a little more complicated now because we want to go from 1 dimension to 2 dimensions. So the trunk of the model is the same as what I've shown you before. We're doing convolutions, batch norm, ReLUs, pooling, dilated convolutions.
And
So
that's all
after
the
this-
representation learning that we've been talking about. So you still have the same representation, and you could even use transfer learning to build on representation that you've already learned from the previous tasks.
Yeah, exactly. So you can even directly plug in parameters that you've learned from these other data. And there's a paper back-to-back with ours where that's what they did. They pre-trained on lots of chromatin data, and then they mapped those parameters over to a new model in a transfer learning setting, and then they tried to predict the high-C. But we found that we can just run it from scratch.
Yep.
Okay, so we're coming in here with a really long sequence. So I went from 1,000 nucleotides to 100,000. Now we're at 1 million. There's 1 million nucleotides coming into this thing. And so we're starting to stress the memory on our GPUs. And we can only really do it because we have some big memory GPUs. And we don't need as many filter channels. So we can reduce that a little bit. But at this point now, we have 1D profile in 2,000 base pair bins across the sequence. Let's say that I ran 100 filters in the previous one. So it would be 2,000 by 100 matrix. So now we need to get to 2D somehow to make predictions. So we tried a bunch of things. You can see some of them here on the next slide. We tried a bunch of things. What worked the best was just to take position I, position J, and take their average. So you can see here, we tried averaging, we tried concatenating, taking a max over each channel, taking a geometric mean, like multiplying and then taking a square root, and taking a dot product, just multiplying, element-wise dot product. So the averaging looks like this. For this filter channel, you come in with this 1D signal, and it's on the x-axis and y-axis, and then the 2D thing is just the average of the two. So if there's, say, something interesting here and something interesting there, then it's going to have a high signal at that position. So now we're in 2D land, and now we can just start to use 2D deep learning tools, so 2D convolutions. So we start just doing, we have the same problem where we need to share information across the sequence quickly. So use dilated convolutions, just in a 2D form. And then because this matrix should be symmetric, and there's nothing in the typical workflows that impose symmetry, the one novel factor we do is every time we do a dilated convolution, we transpose the matrix and take the average between the two. So we re-symmetrize after every layer. And then you make predictions for your data. And it looks like we're about at time, so I'll just say, this works reasonably well. You know, here's some predictions for certain data sets, for targets, and you know, across the genome, it works reasonably well. And CTCF is the main thing it learns. So some of these analyses are kind of digging into how it learns CTCF, what it learns about CTCF, whether it learns anything else. The jury's out on that. CTCF seems to be the main thing. But hopefully the presentation here gives you kind of an overview of, you know, the ways we can think about the data we have in biology and genomics and design neural network architectures that are capable of going from really basic inputs like DNA sequence. You can do this sort of stuff on RNA sequence, on proteins, and with the right set of transformations and enough data, you can learn some pretty cool stuff.
Awesome. Thank you so much, David. This is truly illuminating. So let's see, a quick poll. Who feels that they've learned something? Let's see. Wow, this is outstanding. Good. So we have 71%, 24%, 0, and then one person who's still not excited. So it's okay. Awesome. So thank you so much, David. Thank you all for sticking around a little over time. And we will see you guys on Thursday with more exciting guest lectures. Thank you so much, David. Bye.
Thanks.
Everybody,
Bye.
give David a hand. Thank you.